Good morning, ladies and gentlemen. My name is Rashmi Rajendra, one of the tech support scientists at ACD. Welcome to the webinar on the tips for getting started with RNAscope and BaseScope Assays. A few points of business before we begin this webinar today. The audience will be muted during the course of this webinar. If you would like to ask a question, please use the chat feature to type in your questions. My colleagues are standing by and will be addressing your questions in real time during the course of this webinar and after the webinar. If for some reason we are unable to address your questions during this session, we will definitely reach out to you and follow up with you offline. So the topic for today's webinar is tips for getting started with RNAscope and BaseScope assays. Here's a look at the agenda for today's webinar. We will talk about the RNAscope technology, the RNAscope assay, the types of assay workflow, reagents and requirements, the RNAscope assay results, what they look like, troubleshooting your RNAscope assay, understanding your RNAscope assay results. We'll also talk about the BaseScope assay and brief examples of its applications. And we'll look at some of the applications that RNAscope assay has been used for and some frequently asked questions. So let's get started. What is the RNAscope technology? The RNAscope assay is an in situ hybridization method. It is a slide-based assay intended for use on the same types of samples you would use for either IHC or immunofluorescence, be it FFPE, which is formalin fixed paraffin embedded sections, or fresh frozen sections, fixed frozen tissue sections, or cultured cells. This technology utilizes a unique double-D target probe design followed by a signal amplification system, which will allow you to visualize a single mRNA transcript as punctate dots through either chromogenic or fluorescent detection methods. This system allows for simultaneous signal amplification and background suppression. Here is an overview of the RNAscope assay workflow. The RNAscope methodology involves pre-treating the tissue or cells in order to unmask the target mRNA, hybridizing the target-specific oligonucleotide probes to the target RNA. The target probe is a pool of oligos that have been designed using established bioinformatics algorithms to provide an assay which can detect a single target RNA. Detection is enabled through an amplification procedure that involves sequential hybridization steps to build up a tree-like structure, shown here schematically, which I will describe in more detail in the subsequent slides. With this method, we are able to localize the signal of an individual transcript and can visualize these individual transcripts as dots in a field of cells. Here are the key features of the RNAscope technology. This key feature is a double-Z probe design. Each probe contains 3 elements, the lower region of the Z, an 18 to 25 base pair sequence that is complementary to the target RNA. The upper region is a 14 base tail sequence. A spacer sequence links the 2 components of the probe. The probe always works in pairs as soon as a double Z. The double Z probe contains the target-specific binding site that is a 50 base long, and the upper 28 base pair is the pre-amplifier binding site, and it is where the amplification cascade occurs. With a standard probe design, the target probe set is a pool of 20 ZZ oligo pairs, and it binds to approximately 1 kb RNA target region, as you can see here in the schematic. Let's look a little bit at the RNAscope technology signal amplification. Once the ZZ probe binds to the target sequence, the building of the amplification tree starts with the pre-amplifier and the amplifier binding. Each amplifier can further bind multiple labeled probes. Labeled probes can be either for chromogenic or fluorescent detection. If only one Z binds to the target sequence, the amplification tree cannot be built, and therefore no signal will be detected. This ensures the specificity of the signal amplification. To date, we have over 1,200 peer-reviewed publications using the RNAscope and BaseScope technologies in various applications. This shows the increase in market adoption of the RNAscope and BaseScope assays utilized in many different areas of research. We will talk about some of these applications towards the end of the webinar. Now let's look at what types of assays do we have and what is the workflow for some of these assays. So here is a summary of the assays that are currently available from ACD. We have either brown or red singleplex assay. If you would like to detect more than 2 targets in one assay, you can choose the Duplex or the multiplex fluorescent assays. When choosing the multiplex fluorescent assays, be sure to choose the right fluorophore based on the multispectral imaging and fluorescent microscope capabilities that you have available. The channel designation you see here is not a universal designation, but instead for probes in different channels. If your target of interest is a short sequence or circular RNA, BaseScope would be the right assay for you. The channel designation, and what you can see is that the BaseScope method at the moment is only a chromogenic assay. Also note that some of these assays are available either on the Leica Bond RX or the Ventana Discovery platform for automated assays for the RNAscope assay. Now, once you know the kind of detection that you're going to be performing, the next comes to choose the right probes. We have a catalog of over 13,000 probes at the moment. So the best way to look for them would be on our website at acdbio.com/catalog probes, and you can enter the gene name and search for the catalog probes. They will be listed with the species and the genus and species name of the target that it was generated in. You could also search for the gene name in the search box on our main website. Most of our probes are either were designed for greater than 300 base pairs for regular mRNA or long non-coding RNA, and these are found in the catalog probes. With knockout validations, as we have thousands of probes currently in our inventory, we would like to help ensure that the probe you're picking is specific for your wild type versus knockout validation, in which case it would be a good idea to perhaps contact our technical support department, or you could check in our catalog probes for more information about the probe that you're interested in. If you are working with a novel gene and no catalog probe is available, please feel free to contact your area account manager or go to our website and we can create a made-to-order probe for your research purpose. This is the same method you would use also if you're interested in looking at short sequences, splice variants, circular RNA for 50 to 300 base pairs. For this, you would need the BaseScope probes, which are also made to order. Now, once you know which probe you will be using and which detection assay you will be using, you need to be sure that you use the right protocols when you're using for different sample types. We recommend that you check out our website on our support page. You would look under user manuals, and you can choose the right protocol for your purpose. Most protocols for RNAscope assay are in 2 parts. The first part for sample pre-treatment, and the second part for RNAscope or BaseScope detection assay. Please check out our website for the under technical support for user manuals. If you're working with unique sample types such as whole mount, free floating, or bone, please feel free to contact the technical support for suggestions and guidelines, and we will be able to help you find the best options for your assay. The RNAscope assay can be done in either as a 1-day assay or can be split into 2 days. Here is a schematic just to give you some options as to when you can stop the assay. So briefly, you can either stop the assay after the sample pre-treatment on day 1 and then go on to perform probe hybridization and the RNAscope detection on day 2. Alternatively, you could stop stop on day 1 after probe hybridization by incubating the slides in 5x SSE overnight, and then go on to perform the RNAscope detection assay on day 2. I do recommend that you please check the user manual in detail, where you have all the information and optional stopping points mentioned quite clearly, and this will offer you the right guideline based on your protocol and your assay. Let's look at the workflow now for the RNAscope Chromogenic assay on FFPE sample. This is the protocol overview. As you can see, the main feature, the protocol is split into 4 parts, where you have the pre-treatment, hybridize and amplify, detect and counterstain. The first part, where you have the pre-treatment, this is something that I want to draw your attention to. For FFPE samples, very similar to IHC, you have the bake, dewax, H2O2, and the target retrieval steps, and also the protease digestion. These are the main key elements for a successful RNAscope assay. Also, I just would like to mention that this workflow is very similar to what would be performed on the Leica Bond RX or the Ventana system. Similarly, when you do perform the fluorescent multiplex assay on fresh frozen tissue, again, the pre-treatment procedures are similar, but of course, since you do not have any paraffin, there is no dewax or baking required. You just have a post-fixation step and then go on to dehydrating and protease digestion. But the principle remains the same in terms of sample pre-treatment before you go on to hybridization and amplification for probe detection. The pre-treatment conditions will vary based on the sample type and the thickness of the fresh frozen samples. The RNAscope Multiplex Fluorescent V2 assay has been optimized for FFPE samples. So in this case, again, like in the previous, you do have the pre-treatment, bake, dewax, and target retrieval steps. But one thing that I would like to emphasize is whether you're trying to detect one probe or multiple probes in the same FFPE sample, it is very important to perform all the AMP steps in this assay to be able to detect the 3 different targets. The key feature in this kit is that you would require to buy the TSA plus fluorophores or the opal dyes from PerkinElmer. The information is also available on in our user manual for the V2 assay, or you can contact us at technical support. Let us look at some of the RNAscope assay reagents and requirements to run an assay. Okay, so here are some key guidelines for the RNAscope assay. First of all, I would highly recommend that you please follow the protocols exactly as described in the user manual. These have been optimized for you to achieve the best possible results using RNAscope assay. The second part that I would like to bring your attention to is sample pre-treatment. Please do review the sample pre-treatment conditions for your specific sample type. Always use control probes and slides. This is very important, and I will talk about that in a little bit subsequently in one of the slides. And always review the checklist before you begin the assay. Here's what the RNAscope detection reagents look like. You will receive them in boxes that contain all the reagents that you require to perform the assay. Here is a checklist of some important items that are a must-have for success with RNAscope assays. All this information is also available as a user manual for your future reference. I would like to point out some key elements such as the Superfrost plus slides, either using a hot plate or steamer for pre-treatment and target retrieval, the high-DZ oven, which is very important to maintain the humidity and temperature of your hybridization and amplification steps, and also the use of appropriate positive and negative control probes during the assay. Please note, EcoMount is the mounting medium to be used at the end of the red assay, Vector mount for the Duplex, and Prolonged Gold anti-phase mounting medium for the Fluorescent Multiplex assay. All this information is also available on our website or in the user manuals, or you could ask us at support@acdbio.com. Here are the recommended accessories for the manual assay. You would require the easy batch slide processing. This makes the processing of multiple slides very convenient. As you can see in the image here, this is the Hy-Vee Z2 oven for temperature and humidity control. The TissueTech washing tray for washing the slides. And either you could use the hot plate or hot plate with a beaker, or a steamer for the target retrieval step. Both these options work fairly well. And as you can see in the image, it is recommended to dip the entire set of slides into the target retrieval if you're using the beaker and the hot plate. Here are some of the guidelines while performing the assay. Please perform all the AMP steps in the right order and do not skip them for getting optimum results. Okay? Do not let the slides dry out unless mentioned in the protocol. Make sure you use the hydrophobic barrier pen so that the reagent is so that the samples do not dry out. Please do not alter the protocol in any way, as this will only result in suboptimal assay results. Try and use fresh reagents as much as possible, and please check the protocol and reagents well before starting the assay. So let's take a look at some of the RNAscope assay results. So before you even get started with getting results, you need to qualify your samples using appropriate controls. This is how we would recommend that you qualify your samples for the assay. We always recommend to use a negative control probe, DAP-B, and positive control probe, for example, PPI-B, with your sample type. If the technique check passes with robust signal in positive control and clean background in the negative control, then you can move on to test and run your target probes. One way in which we describe a good path would be if the PPI-B has an ACD score of 2, and I will talk about that in a later slide. And if the DAP-B is clean with no dots and less than 1 dot per 10 cells. In the event that your control probes in your tissue fail, then you may need to optimize your assay conditions. If you pass, then we can recommend to move on to testing your probes. So in these cases, if it fails, we recommend that you verify the technique, check the RNA quality with new samples, and perform some assay optimization for your tissue type and for the specific conditions that you're analyzing. Here are serial sections from human lung cancer samples on which we perform the manual red chromogenic assay. And here is a typical example of how an assay should look like. On the left is a section stained with our negative control probe, DAP-B, which is a bacterial gene that is not expressed in intact tissue. As observed in this sample, it should be no signal at all. Here is the positive control, PPI-B. As you can see, there's uniform staining throughout the sample. Certain tissues may express the positive control probe at a higher or lower level, but in general, you want to see the expression throughout the sample. With these results, knowing that it has worked well, then you can proceed to the checking your target probe. And here is a test probe with PDL1. As you can see, the results are consistent. I'm showing a section from the same tissue sample, serial sections, so you can get an understanding of what these samples would look like. So why would you require to optimize an assay? Why would you optimize your assay? So here are some potential reasons that you may need to optimize your assay. If the samples are underfixed, this can usually lead to over-digestion of your samples using the standard conditions. If you overfix, the signal could be weak and you may need to optimize the samples, optimize the assay. There are also special sample types that could require different pre-treatment conditions and optimizations, with a few listed here, such as xenograft or cultured cells or whole mount, or special tissues such as lymphoid tissues, for example, spleen, tonsil, or lymph node. And if you have any questions regarding the special sample types that you're using or any other issue, you could also email us at support@acdbio.com. Now let's look at some ways in which you can troubleshoot the RNAscope assay. So to begin with, we recommend that you run the assay on your sample and begin with standard conditions of pretreatment, which is 15 minutes of target retrieval at 100 degrees and 30 minutes of protease plus at 40 degrees. This is for FFPE samples. Then you observe the signal intensity and background with the control probes, with your positive and negative probes, and also look at the sample morphology. It's always best to start with standard conditions, and then based on the result that you get, you can optimize, either by adjusting the target retrieval time or by adjusting the protease time. And in some of the slides in this troubleshooting section, we're going to talk about the various ways in which you could do this. These similar optimization methods are also available for optimizing your assay when you're running the assay either on the Leica Bond RX or the Ventana XT or Ultra systems. You can either alter the pretreatment or time or the protease time for either overfixed or underdigested. And in this case, it would be good to contact your local field application scientist, or you can refer to the protocols for pretreatment optimization guidelines. So here is an example of RNAscope results with optimum tissue digestion. Okay? The assay condition was 15 minutes of target retrieval at 95 to 100 degrees Celsius. This was done on FFPE samples with 15 minutes protease at 40 degrees for the specific tissue type that was used. The intact tissue morphology here shows uniform PPIB staining on the left with clean DAP-B staining on the right. And as you can see, homogeneous hematoxylin staining all across the tissue. Now here is a condition in which how the results look if there has been over-digestion, okay? In which case the target retrieval was not optimum. You can see the destroyed tissue morphology, undefined nuclei, and weak hematoxylin staining. So here is a way in which you can overcome the issue of over-digestion. So over-digestion can happen if your samples are underfixed. So these are FFPE samples, which was fixed for 12 hours as opposed to our 24 to 32 hours or 18 to 32 hours. As you can see, the morphology is destroyed. And in these two panels, you can see on the left, you can see the DAP-B staining even shows a few dots. This is background with non-specific staining in the nucleus. The hematoxylin staining is not very strong, and the boiling was done at a standard of 15 minutes. Now, if you reduce the boiling, as you can see on the right panel, the nuclei is stained really well with the hematoxylin, and here there is no DAP-B staining in the background, because the DAP-B should be clean. It's a negative control. Same experiment with the positive controls. On the left, you can see the staining is not very distinct. The hematoxylin is not clear with the PPIB, with the 15-minute boiling. And when you reduce the boiling and the target retrieval conditions, you can see that the staining is more clean and significant. Now let's look at the opposite direction, when the samples are underdigested. The PPIB is much weaker than the first two samples, as you can see. Here, 10-minute target retrieval and 15-minute protease was done. The morphology is excellent, but the PPIB is weak. So in this case, the recommendation would be to modify the pretreatment conditions to get better staining. Again, looking at underdigestion, also with the PPIB staining, you can see that these samples are overfixed. These are 72 hours fixed samples. And you can see the low intensity, the dense compact nuclei, and you can also see background trapping. Now, when you increase if you increase the boiling conditions for the target retrieval, you get better signal resolution. The RNAscope assay can also be optimized for suboptimal fixed samples. For example, here on the right, you can see these samples were fixed for 3 weeks. And we have pre-treatment condition modifications that can improve the signal to get better signal resolution. Now, what about over-digestion for the fluorescent assay? On the left here, you can see fresh frozen mouse kidney and fresh frozen mouse brain samples that were treated with protease 4. They're over-digested. You can see the non-specific staining and a general autofluorescence as well in the tissue. Now, if you optimize the protease pre-treatment at room temperature versus 40 degrees, you can see a better signal detection. This was done with 2 probes. You can see with the fresh frozen mouse kidney on the right panel, and on the right bottom panel, you can see better resolution of the signal. So in this case, there was a decrease in background and an increase in signal. One known issue with the fluorescent assay is due to the long fixation time for FFPE samples. And a lot of FFPE samples are prone to inherent autofluorescence, which poses challenges for image analysis. You can use FFPE samples if you have background correction software to reduce the autofluorescence. And alternatively, we now have developed a multiplex fluorescent assay V2 assay, which can help you address this issue. But even after taking all of this into account, if you are still experiencing autofluorescence issues with your assay, please do contact technical support. Another level of troubleshooting that can come up is when you see no signal or weak signal or non-specific signal. One issue could be just something as simple as the microscopy. Perhaps you're using the wrong filter setting or the longer emission cutoff or perhaps wrong exposure. So it's important to choose the right fluorophore when you're performing RNAscope assay so that and taking into account the multispectral imaging and the microscope conditions that you have. Also, please use the correct filter settings. Do not use auto exposure the very first time. Verify the signal with the naked eye if possible. And if possible, use the known image enhancing software. And if you would like to ask us more questions about this, please do email us. Here are some tips for each of the assays we have available. First, we have the chromogenic brown assay. It is the most robust assay, which is good for the first-time RNAscope user. But we would recommend it specifically if you're working with tissues that may have an inherent alkaline phosphatase-dependent background, such as kidney or adrenal glands. We would not recommend you to use the chromogenic brown assay if you're working with pigmented tissue, such as lungs or skin. As you can see with this panel here, in the mouse, PTIB staining in the kidney, there's uniform staining and with brown dots that you can see all over the tissue. Here's an example of red chromogenic assay. We highly recommend the use of EcoMount as a specific mounting medium. As other mounting mediums may cause red signal diffusion, it is also very important that the red signal stain slides are not exposed to any alcohol, as this will also cause diffusion of and potential loss of signal. For the Duplex chromogenic assay, here is an example of 2 genes co-expressed in FFPE human colon cancer tissue. Again, do not dehydrate the sample with alcohol, as the red and green chromogens are sensitive to ethanol. Here is an example of fresh frozen samples stained with the RNAscope Fluorescent Multiplex Assay. We do recommend using prolonged gold as the mounting medium, as this prevents the loss of signal that you can see. There are 3 options in the AMP4 step that you could utilize to get different color combinations. Here is an example of data using the RNAscope Multiplex Fluorescent V2 assay. This is recommended for FFPE samples, fixed frozen samples, or other samples where the RNA expression is low. Please note that the fluorophores here, the TSA plus or the opal fluorophores that we recommend to be used with this kit need to be purchased from PerkinElmer. Here are some of the factors affecting RNAscope assay performance. When the fixation conditions are not optimal, if the RNA is degraded, the hybridization conditions are not optimal, if the samples dry out during the assay, or if you are using special tissues that are sensitive to pretreatment. And here are some ways around it and some solutions that can help you get good results. Please do fix the samples as recommended. Example, for FFPE, you can use 10% neutral buffered formalin at room temperature for 16 to 32 hours. You can acquire new samples and assess the RNA quality, like I mentioned before, by qualifying with positive and negative controls. Please use the high-BZ hybridization system. This maintains the temperature and the humidity during the course of the assay. Use the image pen and add adequate reagents to avoid drying, and start with standard pretreatment conditions and optimize the conditions accordingly. One of the issues that does occur and that has been observed is the issue of sample detachment. Why would tissue samples detach from slides? What are the possible causes? So one could be the wrong slides were used. And so that's why we recommend using Superfrost Plus slides, as these are charged and help adhering of the tissue to the slide during the whole procedure. Or the tissue wasn't prepared properly. So we do recommend that you please prepare, fix the samples correctly and prepare the slides as per recommendation. And these are all also listed in the protocol. But many times, even with the best conditions, tissue detachment will take place. So we do have some modifications and some suggestions to help you overcome these issues and get optimum good RNAscope results. So one way in which, under certain conditions, FFPE samples might detach during the course of the assay. We have modifications to our protocol where you can incorporate two baking steps, either before the target retrieval and one after the target retrieval, which can help mitigate this issue of tissue detachment. And please feel free to email us and we can discuss this in more detail with you. Similarly, for fixed frozen samples as well, we have an additional air drying step, an increased post-fixation step, and a baking step with fixed frozen samples. And these can help tissue detachment during the course of the RNAscope assay. Similarly, for fresh frozen samples, we recommend increasing the fixation time from 15 minutes to 1 hour and doing a short baking step at 37 degrees Celsius before going on to the RNAscope detection assay. Now let's look at how can you understand your RNAscope data. So here is a schematic of the RNAscope semi-quantitative scoring guidelines in which you can count the dots and choose the scoring criteria based on dot count. We do have a data analysis guide online as shown here on this link at acdbio.com/data analysis guide, in which you can in which it's described more in detail about what our ACD scoring criteria is all about. So for example, a score of 0 is when you see no staining or less than 1 dot per 10 cells. And an optimum staining would be either a score of 2 or 3, where you have about 4 to 9 dots per cell, with none or very few dot clusters, or less than 10% of dot clusters. So these are considered as good, as a good positive stain, a good positive score. What does a dot mean? What is the significance of a dot? And what is the difference between a dot and a cluster? These are some of the questions that you probably have at the moment. And let's take a look at that. So in the chromogenic assay, where you have dots versus clusters, what does it mean? In this image, the RNAscope chromogenic assay has been performed with a red chromogen. The signal appears as numerous punctate red dots of various sizes and intensities, and a few clusters. It is the number of dots and or the clusters that are important rather than the size of the dot or the intensity of the dot. Each punctate dot equals an mRNA molecule. The small faint punctate dot pointed by the orange arrow is one mRNA molecule. The slightly larger, darker red, nicely rounded punctate dot is also one RNA molecule. However, it's a larger size due to more ZZ probes bound to the target molecule. In contrast, a cluster pointed by the black arrow is more oblong with a slightly irregular border and results from overlapping signals from multiple mRNA molecules. Similarly, you will observe this similar thing even with the fluorescent dot versus clusters. As you can see, similar patterns can be seen using this in this image that I show here. The orange arrows point to single mRNA molecules with less ZZ probe pairs bound to them, compared with the gray arrows, which are pointing to a cluster with more irregular rather than round shape, resulting from overlapping signals from multiple mRNA molecules. This was just a brief overview about how we have some data analysis, that you can do data analysis using the RNAscope assay, and we recommend that you check out our website for more information and to go into more detail about a quantification. Let's talk about the BaseScope assay and the overview and application. The BaseScope assay is based on RNAscope technology, but there are several major differences. Here is a general comparison between the two platforms. The RNAscope assay utilizes a probe consisting typically of 20 ZZ pairs for each RNA target, whereas BaseScope can detect as low as 1 ZZ pair or more. The BaseScope probes are designed differently, so they cannot be combined with RNAscope reagents and vice versa. While RNAscope assay is a robust way to detect gene expression of most RNA targets, BaseScope is specifically designed for use for detection of exon junction, short target sequences, point mutations, which I will describe in a little later. RNAscope assay is able to detect up to 3 targets simultaneously, whereas currently the BaseScope is a singleplex and can detect only 1 target at a time. Detection with RNAscope assay can be either chromogenic or fluorescent. Currently, the BaseScope is can be used either for only with the chromogenic, but the fast red detection can be visualized fluorescently as well. RNAscope can be performed manually or using the automation systems, but currently the BaseScope is only available for the manual assay. The assay workflow for BaseScope is similar to RNAscope. The probes are much shorter in length. It may require a stronger pre-treatment due to the additional amplification steps that are involved. And again, as a note, please do not mix RNAscope and BaseScope reagents, as these are not compatible with each other. So here are some of the main differences between the RNAscope assay. The probes are smaller, and due to the increased amplification steps, you do require longer or stronger protease treatment, but the protocol and the workflow is essentially similar. Here are some of the main applications for the BaseScope assay. For example, detecting exon junctions, gene knockout, circular RNA. It's a 1ZZ detection there for splice variants, or for short sequences such as CDR sequence for T-cell receptors, highly homologous gene sequences, gene therapy, pre-microRNA, or point mutation detection, short indels, gene editing, homologs. But at the moment, please note that the point mutation detection by BaseScope is only available through ACD's pharma assay services. Also, the BaseScope has been used in very challenging cases where the RNA was partially degraded or in challenging tissues such as bone. I want to quickly touch upon an example of BaseScope assay. Here is just to give you a snapshot of what the data would look like, and also an example of detection of CDR in Jurkat cells. Complementary determining region of the is a highly variable domain in the T cell receptors, and there's only a short region that is unique between Jurkat CDR3 alpha and CDR3 beta sequences. The BaseScope probes were designed specifically for CDR3 alpha and CDR3 beta, and these can distinguish between alpha expressing or beta expressing chain cell populations. So changing directions a little bit, looking at the applications of the RNAscope assay, here is a brief overview of the various applications that the RNAscope assay has been used for. In the basic sciences with neuroscience, stem cell research, developmental biology. In the case of infectious diseases, the emerging pathogens, pathogenesis, inflammation. In cancer and clinical research in the fields of tumor biology, immuno-oncology, and pathology. And biopharma discovery and development, RNAscope assay has been used in a lot of preclinical models, safety, toxicity, gene therapy. The RNAscope assay can detect any gene expression, long non-coding RNAs, qPCR, secreted proteins, the transcripts of secreted proteins. This can be multiplexed. There are high throughput validation, dual IHC. I want to give you a brief example of an immuno-oncology application. Here is a snapshot of images of co-expression profiles from non-small cell lung carcinomas. And here you can see these various pairs of genes expressions. This was done using the Duplex assay. PD-L1 and PD-L2 are expressed in tumor and stromal regions and are co-expressed in the same cells. So this is to give you an example of one of the immuno-oncology applications using the Duplex assay. Here is an example of looking at the mouse brain using the V2 assay, detection of GABAergic and glutamatergic receptors in the brain, V-GLUT, V-GAT, and V-GLUT2. As you can see, different populations of the cells express these various receptors. And here is, and this assay was done using FFPE samples. Here, going into a little bit more detail, I want to talk to you about the application of RNAscope assay with dual ISH-IHC. The dual RNAscope ISH-IHC workflow. This works very similar to the IHC protocol. One of the most important questions that we always get asked is, can you combine RNAscope ISH with IHC or IF? And the answer is yes. Both procedures do have similar workflow shown here that requires sample fixation, some kind of pretreatment to unmask your RNA or protein, and subsequent detection. The recommended workflow is that you pretreat and permeabilize your tissue or cells first, depending on the tissue, and this may require both a heat-mediated target retrieval as well as protease digestion. You would then complete the RNAscope ISH portion, followed by the IHC staining afterwards. Here are some general recommendations to ensure success with your dual ISH-IHC. First of all dual ISH protocols require optimization. In general, it is recommended to combine a working RNAscope protocol with a working IHC protocol. In addition, work with the antibodies and a protocol that you know that are well established in your tissue samples. Second, it is advisable to perform ISH first, followed by IHC. Third, it is advisable to optimize the IHC assay separately using the RNAscope pretreatment reagent to ensure that your protein can still be detected following RNAscope pretreatment. RNAscope requires the protease digestion to unmask the RNA. So do keep this in mind as you optimize the IHC. And lastly, the dual ISH-IHC assay works better for highly expressed proteins due to the protease treatment that is used during the ISH protocol. We have technical notes available on the support page if you combine ISH with IHC or IF. If you have further questions regarding the ISH-IHC, please do contact technical support. Also note, always combine an optimized RNAscope ISH protocol with an optimized IHC protocol. Here is an example of some data using the dual RNAscope ISH-IF staining. This was done on FFPE samples of non-small cell lung carcinoma using the RNAscope Multiplex Fluorescent V2 assay. As you can see, the IDO was stained using fluorescein, the transcript detection. CD274 was also a transcript detection using RNAscope assay, and CD8 was an antibody using immunofluorescence. So before we go into the FAQs, I do want to mention that if you have any questions, please do contact ACD technical support via email. Or you could also, and if you're doing the automation assays, we have field application scientists who are available in most regions of the world. Also, please do ask about the new user program. Please talk to your area account manager about it. It is a scientific consultation program when you are a new user, and the technical support team helps the scientists through the entire process of getting the RNAscope assay set up and optimized during the course of their new user program. So let's look at some of the most frequently asked questions that we hear. One thing that we hear often is, do I need to run every single AMP reagent when doing the multiplex fluorescent assay? And the answer is yes. Each AMP reagent serves as a building block for the amplification structure of your target mRNA. If any AMPs are missed, this may result in no signal. Another question that comes up often is, what does the C1, C2, and C3 nomenclature mean? This is in terms of the probe nomenclature. So in order to detect multiple mRNAs simultaneously, multiple amplification trees must be built on all these different RNA molecules in your tissue. With that in mind, we must ensure specificity. But how? To the double Z probe design, we have a tail sequence that is added. This is indicative of the channel designator, C1, C2, or C3. A C1 probe has a specific tail sequence that will only allow for binding of the subsequent C1 preamplifier, amplifier, and label probe molecules. So here's an example, just a schematic to show you if you're looking at 3 different targets on a multiplex fluorescent. As you have been seeing some of the data during the course of this webinar, the green C1 probe will have a small tail sequence that will indicate that the preamplifier and amplifiers for that particular channel should be built. Similarly for the orange with the C2 and C3. Here are some more questions that you can take a look, and we have always please feel free to reach out to us. One question that we hear a lot is, can I combine RNAscope and BaseScope assays? No, these assays cannot be combined, as the chemistry between the two are completely different and not compatible. And to end this webinar, I would like to emphasize that please feel free to reach out to us in technical support to ask us any questions with regarding sample pretreatment, sample prep, the correct protocols, the detection, and what reagents you would require. But apart from all that, I would also highly encourage you to use the appropriate controls, positive and negative controls, for your specific tissue type while performing the assay, so you can qualify the assay and be sure that the RNAscope assay is working successfully for you. With that, thank you very much for attending today's webinar. And my colleagues are standing by to address some of your questions. And if you're unable to ask them right now, please feel free to reach out to us at support@acdbio.com. Thank you.